So thanks everyone for coming here to this online portal. So I guess the story that I wanted to tell you about today has been developing in our lab for the past couple of years, and as you may guess, based on the title, Heterochromatin organizes the nucleus. Heterochromatin is going to play kind of starring role in this story, but before I get any further with that, I just want to make sure I thank my amazing collaborators, Natasha Naumova, who did all the high C here, Yana Fedorova and Irina Solovey, who did all the microscopy, Jeff, who was a grad student in the Mirny lab at the time and who helped a lot with the project, and of course, Leonid and Job. And we also have the Fortean Center for 3D Structure and Physics of the Genome to thank, as well as this grant. Without further ado, so if you look at your sort of conventional normal mammalian nucleus, you ask kind of where is the active chromatin and where is the inactive chromatin, what you'll see is a picture like this, where the green stuff, the active chromatin tends to be in the interior of the nucleus, while the inactive chromatin tends to be associated with the lamina on the exterior. So the green is the active stuff, the red is the inactive stuff. You'll notice there's also this blue thing here. So this is a different type of inactive chromatin, constitutive heterochromatin. Specifically, what's being stained for here is the major satellite repeat, which is near the, so this is the mouse cell, this is near the end of the mouse chromosome. It's near the telomere, near the centromere. And so if you look at this under microscopy, this is what you'll see. Of course, over the past decade, there's been a great interest in these chromosome conformation capture techniques. And so if you look at those, what you'll generally see, this is kind of the high C equivalent of that separation between active and inactive chromatin. And so what you see here is sort of a checkerboard pattern, and the interpretation that we have of that is that there's generally two types of chromatin, which we call the A compartment and the B compartment. And it seems, the checkerboard, it seems to indicate that A compartment likes to associate with A compartment, B compartment chromatin likes to associate with B compartment. You will notice that I was talking about three things before, right? There's three colors in the image, but there's only two compartments in the high C map, and that's because the blue stuff is highly repetitive, and so you can't map it in high C. And so the way we quantify this a little is you can take some sort of eigenvector decomposition of this matrix after massaging it a little, and you generally get a track that tells you, oh, here along the genome is the A compartment, here along the genome is the B compartment. And of course, it turns out that the B compartment tends to be, is almost exactly the inactive chromatin, and the A compartment is the active chromatin. So for the purposes of this talk, I'm going to be using U chromatin, active, and A compartment stuff is essentially interchangeable. Let's see, what else is there? Right, and so of course I've also indicated like a little blue tag at the end, because that's kind of where you would imagine the constitutive heterochromatin, which is also called a, I guess the physical circle that you see here is called a chromocenter. And so, yeah, so those are that's kind of the physical picture that we have in mind. So I guess, you see the separation, you'd really like to understand what's driving it, since it has a lot of functional consequences. I mean, obviously active, inactive, that's something you'd like to understand, but also it's correlated with replication timing and stuff like that. So we'd really like to understand how the separation of A and B compartments comes about. And with that in mind, it's really intriguing that there are these examples of what are called inverted nuclei that Irina has been studying for a long time, where this conventional organization is actually under microscopy completely flipped. And so all of the heterochromatin, both types, tends to coalesce in the center of the nucleus, whereas the active U chromatin tends to be on the outside. And so you can also do high C for this, and we'll be talking about this later in the talk. And you see that there is, again, this high C version of a separation between active and inactive, and you see this checkerboard pattern. And what Irina has done to characterize this is that she's found that what takes you from a conventional organization to an inverted organization is the absence of a particular sort of tethering protein called LBR, which in mouse cells interacts with heterochromatin to kind of bind it to the lamina. And she's kind of characterized this, and you can look at her cell papers, and it's a series of really beautiful papers where she found that LBR is essentially the cause for this conventional to inverted switch. And so with this in mind, it's very interesting from a physical perspective because in some ways the inverted nuclei are a simpler system. So if you wanted to understand how compartmentalization comes about, you would actually want to look at a system where there are fewer number of interactions. So without the lamina, this is kind of naturally what happens. And so this kind of leads us to kind of the thrust of my talk, which is that we're going to try and disentangle the roles and the uses of the mechanisms of compartmentalization. So there's kind of two general pictures, right? There's a lamina-centric view of compartmentalization, which says that the lamina plays an important part in segregating active and inactive chromatin. And so for a review of kind of the importance of the lamina, here's a great review from Andy Balmont and Basan Sensel. But of course, this can't be the complete picture because even when you lose the lamina, you still have compartmentalization, if in a different, albeit in a different geometry. And so there's also maybe more chromatin-centric view of the forces driving compartmentalization, and these tend to be sort of, the theoretical perspectives on these tend to be heteropolymer phase separation models, and here are some examples of those. Within that kind of broad framework, there's also sort of perspectives that specifically angle out euchromatin or heterochromatin as being the driving force of this separation between active and inactive chromatin. So euchromatin here, the sort of activity-related clustering of euchromatin has been talked about in a couple papers. This is definitely not exhaustive. On the heterochromatin side, there's been a lot of interest recently in the role of HP1 in forming phase-separated droplets. And so there were two great Nature papers that recently came out last year, and I encourage you to look at those if you haven't already, but you probably have. Anyways, right. So so the thrust kind of the rest of this talk is going to be trying to figure out sort of what is the model, what is a simple mechanism that can explain compartmentalization, and does it, what particular elements of this system does it implicate? Is it heterochromatin? Is it euchromatin? Is the lamina part very important? So that's kind of the general question. So to address this question, we did a high C of a number of different types of cells. So obviously we went on to do high C of the rods since they're this ideal model system for kind of disentangling the roles of the lamina and chromatin. We also looked at non-rod neurons, which you can see have a quite different sort of high C pattern to them. We also looked at thymocytes. This is all in mouse, by the way. We also looked at thymocytes over here on the right. And Irina, and this was from Irina's 2013 paper, we also looked at LBR null thymocytes. So as you can see, when the mice nuclei are not expressing LBR, then you get this kind of interesting partially inverted geometry that is nevertheless very different from the conventional geometry, but not quite as clean as the rods. So just to kind of sum that up, right. So the rods are inverted, they don't have LBR, they are postmitotic. Now, non-rod neurons, they are conventionally organized, but they are bigger than rods. So this potentially might have something to do with the lack of, there's many physiological differences between rods and non-rod neurons, so we can't say anything. It's not particularly good control. The thymocytes are about the same size and maybe are a little physiologically more similar to the rods, but they are dividing, so that is one difference. And the LVR null thymocytes are only partially inverted. They are still dividing and still functional, but and you can read about this in Rena's 2013 paper. And we think that the division is kind of related to the partial inversion, which I can talk about later if you like. Okay, moving along. So that's what we did, and well, that's what Natasha and Yana contributed to. And so when we all got together, we said we'd like to really understand this data and come up with a mechanistic framework that will help us interpret it. And so we wanted to start out with a very general model. So we just said, okay, there seems to be 3 types of chromatin. There's this constitutive heterochromatin that tends to be found in these chromocenters. We're going to call this material C monomers in our polymer model. There's going to be B monomers, which are going to represent heterochromatin, the red stuff, and then A monomers, which are going to represent the active euchromatin. And so we wanted to keep this model very sort of agnostic as to what was going on. So the most general version of this is, one of the most general versions of this is that you say, okay, I have 3 types of constituents to my system. That means I must have 6 types of attractions. And I don't know anything about these 6 types of attractions, but I'm going to make polymers that have a particular sequence of these 3 types of monomers, and they're going to I'm going to confine them to a spherical shell and see what sort of patterns they form. And the particular sequence of monomers is going to be dictated by the sequence of A and B compartments that we find from the experimental data. The way we're going to study this sort of general framework is what we do is for every sort of value of these attractions, we're going to simulate a bunch of these polymers using molecular dynamics. We're going to generate a large ensemble of configurations that that a particular model adopts, and then we're going to compare that ensemble to experimental high-C and imaging. So, of course, with 6 attractions, you sort of have a 6-dimensional space that you need to explore that's kind of large to run any thorough sort of molecular dynamics in. So the first thing that we wanted to do to kind of understand this general framework was to see if we could cut down on the parameter space that we would have to explore. So what we noted was that probably what was most important is not the particular values of these attractions, but rather their relative ordering. Of course, that's still not great because there are 720 possible orderings of these 6 attractions. So what we did was we chose 6 numbers and we permuted them to kind of get 720 representative of each of these possible classes of models. So then for these 720 orderings, we generated our molecular dynamics ensembles. We computed the sort of density of each of the types of monomers as a function of the radial distance, and we compared the peaks of each of those individual distributions to the sort of experimental version of this. And so that's what this scatter plot is. You can see that the experiment is this big white dot here, and you can see that there's a small number of these 720 model classes that actually come close to the experiment, and then there's a large number that kind of just don't do too well. So what does that look like in a little more detail? So what we do here is we then just we rank all of the models as a function of how well they do compared to microscopy. And we find that there's sort of a clear 8 models that do really well, and these are the top 8 here. And then I've just kind of highlighted some of the bad models because we also learned a lot from looking at those. For example, if you have a really high interaction between constitutive heterochromatin, the blue stuff, and facultative heterochromatin, the red stuff, then what you get is, of course, mixing between the two. And that's not good because in microscopy, you see a very clearly defined border. Similarly, if you have too low, if this parameter value is too low, then what you have is that the chromocenter, this blue chunk in the middle that you see of the inverted nucleus, gets expelled to the exterior. And so that is not a viable sort of model class either. If you have a particularly high AA attraction, so this is an interaction between active chromatin, what you have is the formation of a large central euchromatin cluster, which is completely the opposite of what you'd like to see. So that kind of underscored to us the commonalities of the top 8 models. These are the average. On the bottom right, you can see that these are kind of the average best parameters for these models, and you can see that generally going from A to C, there's an ascending order in terms of the strength that you want those interactions to have. So you want A to be the weakest and C to be the strongest. We kind of focused in on the best model, which exactly displays this ordering, and so we were going to study that. So that's kind of what we'll focus on for the rest of the talk. But before I go on, I just wanted to note that as a mini conclusion, what we found was that euchromatic interactions must be weak in order to reproduce the inverted morphology. So this is kind of the first step towards that goal we were talking about of disentangling what's going on, what the particular mechanisms are responsible for driving nuclear organization. And so we find that euchromatin can't really be responsible for that. So in the models that we're going to study currently, this is the ordering that we have to follow in order to reproduce the inverted morphology. We're going to simplify it a little further by fixing the CC attraction just to be the strongest. It's just going to be fixed at a level at which we can see a chromocenter, and we're not going to touch it anymore. Since AA must be weak, we're just going to fix it to 0. So that kind of isolates BB, the interaction between the red stuff in microscopy, so the facultative heterochromatin, as the important variable to study. The couplings we just fixed to the geometric mean. This ensures that we have appropriate phase segregation, and this kind of comes from a naive application of the Flory theory of phase separation, which I'm happy to talk about more if you'd like. But yes, so this so for now, we're just going to study a model in which we fixed everything except BB, and we're going to vary BB. And what we're going to do is we want to quantify the, so we did high C, so we'd like to see if we can, by varying BB, we can get our model to agree with both microscopy and high C. So in order to compare our model to high C, we need to quantify it somehow. So I talked about kind of eigenvector decomposition and assigning A and B to particular loci along the genome. So once you've done that and you have an assignment of A and B, then you can take that assignment and you can use that to compute a compartment strength, which is essentially just the sum of same compartment contacts divided by the total number of contacts that you have. So when you do that analysis on our different types of chromatin, what you see is that rods and wild-type thymocytes and LBR null thymocytes have a decently high compartment strength, whereas neurons are a little low. And this is a little surprising because the rods, the LBR null thymocytes, and the wild-type thymocytes all have very different sort of nuclear morphologies, at least as it comes to, at least as far as the distance of heterochromatin to the lamina is concerned. And so we'll come back to this later. But for now, we're just going to focus on varying the BB parameter and seeing if we can get it to match high C and microscopy in our model. So this is just that comparison. What you see here along the x-axis is we're increasing the BB attraction from 0 to 1 kT. And as we do that, we find that we pass through a particular regime in BB space where our model can, in fact, reproduce the same compartment strength that's observed in high C. And in fact, at the same time, the agreement with microscopy is good. Here are the kind of some representative visualizations of what's going on at particular spots in BB space. So this is at the optimal BB. This is kind of what a configuration looks like, and this is what a simulated high C map looks like. And so we think the visual agreement is pretty good. So kind of a mini conclusion from that is that we have that a heterochromatin-dominated model quantitatively explains both the high C and the microscopy of inverted nuclei. So we like that. We feel like we've found a simple mechanistic explanation of what is driving compartmentalization in inverted nuclei. So returning to this point that I was talking about earlier about how sort of strangely similar the compartment strength is across different geometries, we wanted to focus on trying to see if we could then develop our model for the inverted nucleus to a model for the conventional nucleus while still somehow maintaining a very similar sort of compartmentalization strength, even though the geometry should sort of dramatically change going from inverted to conventional. And so the way we did that is we just kind of took the inverted model and then we added interactions of B chromatin to the lamina and C chromatin to the lamina as well. This is kind of in accordance with what Irina has found about conventional and inverted nuclei. And so when you simulate that model, you can do a very similar thing where we did now. Of course, there are two interesting variables. There's the B lamina attraction and the BB attraction as well. And if you sweep both of those, what you find is that there's a spot in this parameter space where both microscopy and high C agree with the model. But these blue lines here are the these blue lines here represent the optimal BB range that was taken from the inverted model for the rods. And so if we kind of parsimoniously assume that the BB attraction is going to be the same in both conventional and inverted nuclei, it identifies one particular point in this parameter space that we should be looking at. And so this optimal point is shown here. And again, we think we have pretty good visual agreement with what's going on, though this is not a... This should be a conventional nuclei, so I apologize for that. Okay, so the mini conclusion is that the nuclear lamina and heterochromatin work together to establish the conventional organization. I saw that Brock said that I have 3 minutes left, so I just want to quickly go through my last slides. And so we also were curious to see what would happen if we could, what would happen if we looked at the transition from conventional to inverted. And what we saw there was that, interestingly enough, I'm just going to focus on the visual similarity, is that if you track what's going on in live cells, so if you track what's going on in real cells and compare it to what we're seeing in simulation, you see that the kind of morphologies that we pass through are very similar. And so that was really interesting to us. And just as kind of a last fun thing to look at, we made some videos. There's a bunch of them that you can focus on. What you're going to see is that the nuclei will kind of jiggle around in the conventional organization, and at some point we're going to turn off the interaction of the lamina with the heterochromatin, and so what you'll see is that the inverted nucleus kind of emerges from that. You can focus on any of them. I suggest this one in the second column, the second to last column in the middle. But yeah, so this is when they're still kind of, we're simulating our conventional model. And at some point right about now, so they've lost interactions with the lamina, and they're kind of jiggling, and eventually they come together into kind of a central chromocenter, though this process can actually take quite a bit of time. So I think just to sum up, as a general principle, what we are hoping you'll be convinced of after this, as a general principle, compartments are forming through the short-range interactions between heterochromatic loci. There's a particular ordering to this, which says that the constitutive heterochromatin interaction must be the strongest, followed by kind of the facultative heterochromatin interactions, and then the euchromatin interactions must be the weakest in order for this model to kind of make sense. And then the other interesting thing that we learned from looking at the conventional model was that, so certainly the interaction between the heterochromatic loci and the lamina are necessary in order to get the spatial positioning of compartments along the exterior of the nucleus, but that's kind of it. You know, it doesn't change the strength of them compartmentalization at all. It really just changes the location of them. So really, we think that the interactions between heterochromatic loci are what's driving compartmentalization, whereas the lamina is simply there to position them in a different place. And so the inverted nucleus is also, in some sense, then the default is the default organization of the nucleus, which then I suppose raises the question of why, you know, most cells would spend the effort of bringing the heterochromatin to the exterior when inverted nuclei seem to be perfectly functional. So with that, I'd like to thank all of the people who were indispensable in making this happen, and of course, the various sources of funding. And we also, if this was too fast and confusing, we also have a bioarchive up that you can look at. So I encourage you to do so. And thank you for your time.